then it turns out to have some kind of fluid between it. So that kind of covers that now. And then, I mentioned when we look at these guys, looking at it from the side is the way we can tell height and gives us a good picture. Looking at it from the top, they're all going to look sort of the same. So the next guy you have is simple cuboidal epithelium. And with this one, as the name implies, it's a single layer of cuboidal cells. So when you take a look at this picture down here on the bottom of this picture up here, they did a really nice job finding cells that have kind of a cube sort of shape. So when you take a look at these guys and they're saying that they're cuboidal, remember that the name is kind of representing some kind of shape. And when you look at it under the microscope, this is what these early guys saw. And they go, oh, it looks sort of like a cube, we'll call it cuboidal. What happens with students sometimes is they come in and go, it doesn't look exactly like a cube. Duh. You know, it's in a human being, it probably won't. Also keep in mind that when you get tissues from a human being, you also have to prepare the tissues, and that probably alters their shape just a little bit. But if it's a cuboidal cell, this one happens to be in your kidney, it has fluid going through it, so the tube gets bigger and gets smaller, and then you lose weight and you gain weight, and you get muscle and you lose muscle, and you change your body shape, you do all kinds of nasty things to your body in terms of you banging around and all that kind of stuff, and some of those cells are just going to get out of shape a little bit. So you just kind of have to use a little imagination and be a little generous sometimes with the names. So nice single layer, those as you can see here. Cube-shaped cell, nuclei are pretty much sort of in the middle of the cell that's present here. And when we take a look at these guys, one of the things we're going to notice is when you see ducts or tubes, cuboidal cells are going to be found there so many times. So whenever I look at a duct or a tube, I usually expect it to have a cuboidal cell that's going to be present. Sometimes they do, sometimes they don't. Most of the time, they actually do. So they have a tendency to line these ducts and tubes so fluid can move through. Some absorptive capacity. In this case, in the kidneys, what will happen is these guys that are not showing it here will, at least in the first part of this structure called nephron. In the first part of this nephron, you'll see quite extensive microvilli that will also be present here to help with that absorptive capacity. When you get down here towards the end of this tube, not as nearly as many microvilli, and we'll also have secretion that will occur. So we're going to move, stuff will move from here, from the outside of the tube to the cell into the tube in order to be secreted. When you talk about these guys in 233, interestingly enough, we're going to talk about tall cuboidal cells and short cuboidal cells. And so they're actually going to be basically cubes, but one group of cells are taller than the other one, so they decided to call them tall cubes and short cubes. And so even though they're cubes, we're going to differentiate there also. And so they're showing you glands, ducts, portions of the kidney, tubules, thyroid gland, all good places for these guys. This is simple columnar epithelium, and when we see columnar epithelium quite often, we are going to see microvilli associated with them. This is by far the dominant epithelial tissue in your GI tract. So as we move through the mouth and we move through the esophagus, we're going to have stratified squamous epithelium. We're going to go through a small sphincter muscle called the cardiac sphincter or lower esophageal sphincter muscle. We go into our stomach, and from that point on, as we go down, it'll be simple columnar, simple columnar, simple columnar, simple columnar, all the way till you get to right towards the end of your rectum and out into your anal canal, and then stratified squamous epithelium begins to take over because there's going to be a lot of friction as you eliminate the parts up there. And so one of the jobs then of the GI tract is going to be absorption. And so when we get to the small intestine, for sure, and then more limited in the large intestine, huge numbers of microvilli, because microvilli increase the surface area. So you can increase the surface area without increasing the mass of cells. Does that make sense? Okay. So if we So here's the columnar cell, and this is the top of our cell. That's the surface area where we can bring things across the top of the cell. We all right? Okay. So what happens then to the surface area? I do this. A little bit or a lot?
A lot.
Yeah, that increased it a whole lot just by doing that. So that's the purpose of the microvilli, to increase the surface area. So it says lining of the stomach, intestines, gallbladder, uterine tubes, and collecting ducts of the kidneys. And when they say collecting ducts of the kidneys, those tubes that we saw before, those were not collecting ducts. Those are called nephron tubules, and they lead to another big tube that's called a collecting duct. Okay? And when we take a look at these guys, one of the things we'll notice quite often with the columnar epithelium is they're going to be held really, really tightly together. And so in your intestines, what's going to happen is this is the lumen of the intestine. This is where our food is. So we're breaking down our food, and we're going to break it down into 4 primary components. Let's say 3 nutritional components. What are they? Lipids, fats, or lipids are fats. Lipids, so go as one big group. Carbohydrates, and proteins, and then nucleic acids also, the zip for nutritional value. And so what we want is to bring those substances from here into here. Are we okay? But you were eating that sandwich today, and you intended to wash your hands, but maybe you didn't. And then you sat down at the table, and people were walking by talking, and the aerosols went out of their mouth and landed on your sandwich. And so as you were eating your sandwich, besides carbohydrates, lipids, and the proteins that you had, you also got some bacteria and some viruses, and who, Lord knows what all, fungal spores, and all kinds of cool things. And do we want them to go inside our body?
No.
No, because we're a tube within a tube, so technically between your lips and your anus, you still are not internal, even though you are inside. Make sense? Did I lose you? Remember, epithelial, right? Epithelial right here, right? Epithelia way down there, epithelia all the way in, connecting to the epithelia back here. Right? A tube within a tube. So between that epithelium and this one, that's internal. So, but that tube happens to be inside, but technically anything in that tube is not internal. Are we all right? Okay. So we don't want that stuff to go from here to here. Germs inside your body are not good. So we take these cells and we hold them together with tight junctions. So what happens is in your GI tract, you do not move material between the cells. You absorb the material through the cells. We actually have to bring it through this membrane, through this cell, and then out this membrane over here. So structure is related to function. If I have 23 layers of this, how fast is absorption going to occur? Not very fast, which means the food is just going to slowly move through the GI tract, right? And so that's not going to work well. And so this is the simplest method of absorption, was to have one layer, correct? Okay, but we wanted to have a little bit of protection and control, so what do we do to the size of the cell? As tall as it can be, as far as just a simple cell. Are we okay? So I made a tall, thick cell. I stuck it together so nothing could get in between, and I made one layer so we can have reasonably fast absorption from the GI tract. Make sense? Cool.
Can I ask a question? Where are the goblet cells in relation to all of that?
In this picture, they don't even show the goblet cells. So what happens is goblet cells would just be between these guys. And then where you saw a goblet cell and the adjoining columnar cells, they would be held together by tight.
And their purpose is to help with absorption or to
produce mucus.
And that helps? Is that to be helpful for some reason or just?
Yeah, because if we didn't produce mucus, don't hold me to this one, because I read this a while back and I'm going to figure out where I saw it. But at one point I read something that said if you didn't produce mucus and saliva in your mouth and your esophagus, it would take you about 3 to 5 gallons of water just to swallow your food for the normal meal. Because that's how a great lubricator it is. So what happens then, in our GI tract, you have this layer of mucus that's here. Now that's there for several reasons.
So the mucus, the goblet cells are going in that direction, not?
Yeah, so if you look at a goblet cell, it's called When you look at it under the microscope, he's a little deceiving in terms of
Kind
what
of
he
like the
looks
top
like.
of a volcano on that section.
Yeah, but because of the way the stuff stains, and some stains well and some doesn't, he's actually sort of a columnar looking cell, but he's got this gigantic vacuole that's here. And so what happens is that he secretes mucus out here, and he kind of narrows down here this big nucleus here, and so it gives him a goblet appearance. But this is all mucus. It goes up here and lines up here. Are we okay? And then you have some glands that have ducts, and ducts would probably be lined with what? Simple cuboidal epithelium, and these are called mucus glands, and they secrete mucus up here also. So it does two things. One, or actually more than two, it lubricates the stuffal flow. Two, if you're a germ, or bacteria here, has trouble getting through the mucus. Is everybody with me? Three, these guys right here are secreting enzymes, and they go out here, and what happens is the enzymes are activated, and then the enzymes break down your food. So if I've got this mucus layer here, and the enzymes are activated so they get out here, then the mucus actually helps the enzymes not come in contact with my cell, because if I just ate a piece of turkey, the same cells, the components of turkey, are the same components of these cells right here. So our, if we didn't have the mucus, our digestive enzymes would eat our GI tract out. That's not a good thing. Are we okay? Does that make sense?
I have a question. So are microvilli and cilium only on the columnar epithelium?
No, actually, we can microvilli, like I said, you can find them on cuboidal epithelium, for example, on the kidneys. And then cilia, we can have any place we want to move things along. So
Okay.
they won't necessarily only be associated with columnar cells. But when we see columnar cells, quite often, they're going to have microvilli on them in a lot of places in the body. All right, so up to here, everything's been real simple. Right? Pretend like it's been real simple, I agree with it. So now it's going to get a little complicated. So these are actually, this is an example of a simple tissue. So by definition, a simple tissue has to have one layer, and all the cells that you're seeing in this picture are all attached to the common basement membrane, and the cells are not stacked on top of each other. What happens in this particular tissue, so this is pseudostratified ciliated columnar epithelium, is that this is so compacted and it is so dense that these guys are pushing and squeezing on each other like crazy. And what happens is they push on each other to a point where this guy's nucleus goes down and he's actually behind these two cells coming up, and this guy's nucleus gets pushed up, and the bottom of this guy's behind this guy and he's going down, and they get all pushed around. So when we look at it, and we just look at the nucleus that are here, the nuclei that are here, it looks like there's multiple layers of cells because we have multiple layers of nuclei. But the reality is it's one layer of cells, but they're so tightly packed together that some of the nuclei got pushed down and some of them got pushed up because it's too tight to lay them side by side.
Kind of like a log jam.
What?
So it reminds like a log jam, as you start.
Yeah, kind of like the log jam, exactly. This guy will quite often be associated with goblet cells. And when we take a look at this guy, most of the time, we will see this guy associated with cilia. So, kind of when we talk about it, we call them pseudostratified cilia columnar epithelium. It's just a habit, because cilia is almost always present. So, when I show you up here, it's just lining of the nasal cavity, the trachea, the bronchitic portions of the male reproductive tract, and it says functions are protection, secretion, and move mucus with the cilia. So the big function for this guy is going to be the protective function, and part of the protective function has to do with the fact that he produces mucus. So one of the big, huge roles of this particular tissue is when we take a look at the reproductive system, starting with the trachea, going all the way down, way deep into the lungs, we find this guy, and what will happen is these little cilia are going to constantly wave, beat in this rhythmic wave-like fashion. And what happens is cilia are much more complex and better coordinated than flagella are. So we can get these guys to start waving, and then the guys behind them start waving, and they can actually keep a fluid moving or some other structure moving in a particular direction. And that's what their job is. They actually make what we call a respiratory escalator, and that they constantly move mucus upward, 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 up through your trachea. And then right up here, your trachea and your esophagus come together, right about there. And so it gets to the top of the trachea, and then when you swallow, it slides down the esophagus, and all that mucus goes into your stomach. So the job here then is to keep material that you breathe in from getting deep in the lungs and staying in the lungs. So we brush it up, we brush it up, we brush it up, we brush it up. Does that make sense? Okay. And pseudo-stratified, pseudo means false. So it looks stratified, so it's falsely stratified. So when we take a look at this guy's function, here's our smoker, and this is the normal respiratory epithelium. Little particles come in, and one of the things we find with cigarette smoke, there's huge amounts of particulate matter. And then the other thing we find is some of the chemicals that we have in cigarette smokes actually inhibits the action of these cells, causing them to be less active, so we have less cilia moving every time that you're smoking a cigarette. So if you do that day in and day out, and day in and day out, what will happen is the particulate matter is going to land on that mucus, going to make it thicker and thicker and thicker, affect the ability of those cells to keep moving that mucus forward, forward, forward. And eventually what can happen is those chemicals can start to affect the cells inside of the lungs and the esophagus and the trachea. And so if you look at the top up here, we have these guys being paralyzed, and mucus begins to get built up, and some of the cells begin to change, and they no longer have cilia, and so that's called dysplasia. And then over in the next picture, what's happening is these cells are now starting to grow, and they're not actually replacing themselves with normal pseudostratified ciliated cells, and the mucus builds up, and we have less of a mucus elevator, and so it's really starting to change drastically. And if this continues with this process, then we end up in this state, which is called anaplasia. And what you have here are these cells are not going anywhere close to what their normal job should be, and now they're growing uncontrollably. They don't listen to any kind of signal that says stop growing. They'll start to push on these cells over here. These guys, because they're crowded and they're normal cells, they will stop growing. They'll be replaced with these cells, and the tumor gets bigger and 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 bigger. And you get a serious case of lung cancer or whatever kind of cancer here. That makes sense? Are you okay? I'm not going to ask you to smoke, but I'm going to tell you the number one thing they tell you is to be a healthy person, is if you smoke, that you stop smoking. Are you okay? And so I will warn you right now. I have a few soapboxes. I get on my soapboxes every once in a while. When I get on my soapbox, I will tell you I'm on my soapbox. I'm on my soapbox. Okay, so don't smoke. It's bad for you. It's bad for the people around you. And we're all freaked out about the cost of this and the cost of that. If you could just get all the smokers to quit smoking, the cost associated with care for smokers and lung cancer and all that would drive health costs way down. It's a really expensive proposition, and 80% of lung cancers are directly associated with smoking.
Does vaporizing do the same thing as burning?
In terms of those
This
electric
process right here?
So when you vaporize,
When you inhale a vapor,
Turns
it's the same.
Oh, it would depend on.
If it can burn the material.
Well, yeah, it would depend on what's in the vapor. Yeah.
I know like this e-cigarette, I mean, it's.
Yeah, I don't know what's inside of those, if it's the same stuff as tobacco and things, but if you have the same chemicals that will be present, they're part of the release of the chemicals is the burning process. So if you had the same kinds of chemicals that were in there, it would do the same thing, but I don't know if that vapor has all those chemicals in it. Are we okay so far? All right. And for those of you who do the other kind of smoking, don't tell me it's organic and it's okay. This is also a plant, and you're burning it just the same, and so it doesn't have exactly the same chemicals, but it's also not for people who get for your lungs. Are you with me? Yeah, I'm off my soapbox now. And I'm not, I just, I will warn you right now, I'm not trying to be offensive if you're a smoker, I just would really love for you to stop so you can be healthier. Every time I can talk about smoking, if they stop, I'm gonna do that for the next 3 terms, so I'll just warn you sometime. Alright, so, and just think, $5 a pop for those guys too, how much money is it? And so stratified squamous epithelium, stratified means?
Layers.
More than one layer, squamous means? Flat cells. When we look at this tissue under the microscope, one of the things that we notice is not all the cells are flat. So this is part of that polarity we were talking about last time. So this is stratified squamous epithelium, and we see really flat cells up here. And when we take a look at the numbers of layers, it's dominated by these squamous cells. Even though this layer up here doesn't appear super thick, maybe compared to the spinosum down here, what happens is that the reason that it doesn't appear super thick is because they're flat. But there's about 25 to 30 or more layers of these flat cells up here, and down here there's probably around 12 to 20 maybe of some of these other cells. So this guy really has a lot of these flat cells that are present here. So location, it says surface of the skin, so entire cutaneous layer of your body is stratified squamous epithelium. Lining of the mouth, throat, esophagus, we already talked about that, but it's for continuation. Out here, this is called keratinized stratified squamous epithelium. Inside your mouth, it is non-keratinized. In the next chapter, we're going to talk about what that means. And then we go down through the rest of our GI tract here, which we said was,
Colon.
what kind of colon?
Squeal.
No, not,
Something?
no. Simple columnar or GI tract. Did they say respiratory tract? Or say GI?
Yeah.
GI tract, okay. And then we get to where the friction starts again. We're back to stratified squamous epithelium. So rectum, anus, and not part of the GI tract at all, but vagina, because there's a lot of friction that goes on there also. All right, does that make sense? Okay. So big function then is probably protection, exactly. The big function here is going to be protection. So protection from abrasion, and then also from pathogens getting in, and all that good stuff. Now what happens is, when we take a look down here, these guys are more cube-shaped. So what we see down here are a group of cells that are called germinal cells. And the germinal cells divide in mitosis and push upward, and that upward movement continually pushes this layer of cells that's falling off all the time. Questions about that, guys?
So then, if it's cuboidal at the bottom and it works itself out, does it change to being squamous then?
Drastically, yeah. So what happens is, when they named it for the upper layer, because epithelium is dominated by a free edge, and the fact that this, when you take a look at the numbers of layers, the number of squamous layers is way outweighs the number of any of these other guys. And so there's a process as you go from here to here called keratinization, and we're actually going to not only change the shape of these cells, we're going to change the entire chemical composition of the inside of the cell. So by the time it gets up to here, literally what you have is a phospholipid bilayer and basically which is a fat for keratin. And keratin is a really tough, cornified, waterproofing material, and that's what gives that outer layer of your skin its characteristics. Did that answer your question? Okay. Any other questions? And when we get to the skin, we'll go through all this whole process. So we won't spend too much time. All right, so this is stratified cuboidal epithelium, and this guy is really, really rare. In one place I worked, there was a guy that was a doctor, and his specialty was tissues, and he did all kinds of pathology stuff. He lived in South America for a really long time. And if you wanted an argument, just mention that you saw stratified cuboidal and a couple other kinds of tissues that you argued were not there because they were actually transitional tissues. Transitional meaning they were changing from one kind to the other, and you just couldn't stop and start, so you thought they were changing, which is kind of one of the fields of tissue gases they kind of argue about. So anyway, when you see these guys, protection, secretion, absorption, just like in most ducts and tubes. And so this guy that you're seeing right here, this is a sweat gland. They're also called pseudoviviparous glands, if you see that term also. And this is basically, we talk about these guys being present in the body, this is where we talk about it. And that's just about it. So won't spend a whole lot of time with this guy. Transitional epithelium, another one of those guys that's a little bit different in that when you take a look at him, he doesn't always look the same. So the role of transitional epithelium is to transition. It's actually going to change shape. It needs to change as the organ that is associated with actually changes. So what you notice is this locations, urinary bladder, renal pelvis, and the ureters. So this is our urinary bladder right here. These guys coming up, these two tubes, these are called the ureters. And then we get up above here where our kidneys are at. And the kidney has this sort of hollow region where the ureters are attached. And the ureters are going to be collecting fluid. And so they go inside here a little bit. And this is our renal pelvis right here. And so at the beginning of the urethra is part of the kidney. Excuse me, ureters is part of the kidney. So what happens is this part fills up with urine, this part fills up with urine and stretches. Obviously our urinary bladder fills up with urine and stretches. So it's called transitional epithelium because it transitions from being really tall epithelium with sort of narrowish looking cells to really short epithelium with broader cells. And so in this case, we have an empty bladder and a full bladder, and they have two different appearances. So usually when we see these guys in the lab, what we do is we see them in a relaxed state. So they look really sort of like sometimes, especially the top cells, like somebody took some round river rocks and started stacking on top of each other to make a wall or something like that. And then as they go down like this, if the bladder is bigger, they get a much smaller profile, and then they get wider. Okay? Once again, relegated to the urinary system because they need to have that stretching that goes on so that the bladder will fill up and you won't be ready for the bathroom all the time. Or have to wear pants or something. So we also have glandular epithelium. And so in this picture, they're showing a goblet cell, a nice big blow up. Here's our nucleus. There's that big vacuole. Vacuoles aren't common in animals. And vacuoles are very common in plant cells, probably learned that to 11. So this is kind of an unusual thing to have this big vacuole that's present here. So this guy is going to be secreting a mucus, and so he's going to be called a gland. So if you take a look in your note on page, it says glandular epithelium, and it says extracellular gland, and one of the cells that secrete a particular product, and so it says unicellular extracellular gland, and an example of that guy is a goblet cell. So uni means one. So it's a gland that's actually made up of one cell. And it has to be a gland because he secretes. So it's a unicellular extracellular. So that's our goblet cell. And what's that tissue associated with? Simple columnar epithelium. Very good. Any questions about that guy? Okay. On the next part of the notes, so we'd be on B where it says multicellular extracellular gland. Multicellular, what does that mean? It has to have more than one kind of cell. So when we take a look at these guys, we're going to classify them according to their product and how they secrete their products. And we're also going to classify them according to what they look like. So in your notes for where it says means of secretory discharge, that's what we're going to talk about first. And so when we take a look at the top, this should, I hope, look familiar to everybody. We have the nucleus that's there, right? And then the nucleus produces RNA, and the RNA goes out to the ribosomes, and the ribosomes make proteins, and the proteins tell our cells what to do. And so once we make all of these proteins, then we can start producing stuff, and then we can start storing stuff. So we have our smooth and rough endoplasmic reticulum, and the rough ER we make proteins, and the smooth ER we modify lipids and carbohydrates and things, and we produce all these guys. And then what we do is we move them to the Golgi apparatus where we store them. And so if we need to secrete something, we're going to secrete the stored in that Golgi apparatus. And then we pinch off a little piece because it's a big membrane sac, and that little piece is called a vesicle. It moves up to the surface of the cell, and it just blends with the cell membrane, and whatever's inside that vesicle goes out. Some people go like this, and some people go like that. So does everybody start to remember that until 11? Okay, so that process until 11 we simply call exocytosis, just moving stuff out of the cell, and it's a very kind of simple, straightforward process. So, what we find is when we take a look at some of the glands in our body, this is how they release their secretions. So, let's go. We're going to do a pretend thing here for a second. Let's pretend this was a secretory cell right here, and he was going to secrete stuff into a duct, but let's pretend that he's going to do it through exocytosis. And so what happens is that this guy would produce these little vesicles that move to here, they get emptied out into this little lumen, and this lumen would eventually lead to the surface of our skin, and so we would call him a Marroquin gland. You're right? Because the Marroquin glands are going to secrete their products through exocytosis. So if we say Marroquin gland, no matter what gland we're talking about in our body, the secretions were being released by exocytosis. Does that make sense? All right. The other kind of gland that we have is going to, and so in this case, the salivary gland is a good example of a Marroquin gland. This is called apocrine secretion. So these glands are called apocrine glands because of the way that they release their secretions. So in this case, what's happening in the mammary glands, the cells that are there are going to do the same process. We're going to make the substances we want to release. We make the little vesicles right here. So these are secretory vesicles. And up here, what happens, these guys came to the surface, and then they went through the surface of the cell, all right? Okay. In this case, what will happen is these guys are going to come to the surface of the cell, and they're going to stop. And then the cell's going to pinch in, pinch in, pinch in, pinch in, making the membrane across here. This piece is going to be released, and this cell's going to be left over. And what he's going to do is he's going to keep growing, and then repeat the whole process all over again. So that's called an apocrine secretion. Are we all right? Okay. So any gland that releases its product using this method is called an apocrine gland. And so we're going to give you an example here of the mammary gland. Now, structure is related to function. And so what happens in this case is one of the reasons that this guy probably does what he does is the mammary glands are responsible for producing the nutrients for babies. And one of the things that you want is, because of the way the baby's GI tract is kind of put together, he doesn't absorb some things as well as you do, and he absorbs other things better than you do, and they seem to be able to absorb a little bit larger molecules than we can, and that means they can absorb amino acids, not amino acids, antibodies, proteins. And so what happens is, by housing them like this, it kind of means that as we move through the GI tract, everything gets broken out at the proper rate so that we can release some of the products into the intestines, and then the baby can absorb these larger molecules that haven't been broken down yet. And so that's one of the ideas behind this one. This one down here, this is called a holocrine secretion, and so the glands that do this are called holocrine glands. The oil glands of your skin actually secrete this way. And what happens here is the cells simply go through mitosis, and they move upward, they produce their secretion, and when the cells get to the top, so the edge of this gland right here where the lumen is, they simply just rupture. And so when they rupture, the entire cell's contents get released, and when the contents get released, whatever we were producing also gets released. So what we find with these guys quite often is the secretory vesicles are going to be really, really large, and there's a large number of them, and they have a tendency to dominate a lot of the cells. So when the cell blows up, release a lot of that stuff out. Does that make sense? Okay. So if I release my secretions this way, I would be called a holocrine gland. So all you're doing when you're doing reading now is they'll start talking about a gland being a American gland, or a holocrine gland, or an African gland, in different systems we're going to look at. And all of that's going to tell you is that's how they release their secretions, and that's how they get named.
Where are holocrine glands?
So holocrine glands are like the oil glands on your, yeah, on your skin. Yeah, so let's talk about these guys a little bit. What happens is their job of these guys is predominantly to release oils and some lipid kinds of products. Are you with me? All right. And so when you get to puberty, these glands are really drastically affected by hormones. And so the production goes way, way up. And so what will happen then is that because the production goes way, way up, you get more and more and more and more oil inside of here. And we have some bacteria that also like to live in our skin and in here. And because these are whole cells that are blasting apart, sometimes stuff gets caught up here. And so if you leave oil out for a while, what does it do? It goes rancid. And that rancid is actually oxidation. That's the name of the chemical process. And so when this oil oxidizes, it turns kind of a blackish gray color. And so if it sits there long enough right at the edge of the skin, and it starts to build up because it can't get out because it's a little clogged, we end up with a blackhead, right. And if it sits there long enough, and the bacteria really start growing like crazy, and we get a little inflammation going on, then we're going to build up some pus, and we get zit. That really, it's the one that's always guaranteed to show up on prom or your wedding or your first date with somebody that's really cute, or those things. Does that make sense? Are we okay? And so one of the ways that we have to deal with that is to try to use things that will help wash the oils away, but we can't use anything that dries out the skin. Because if we dry out the skin, what will the skin do? Produce more oil. And so we got to do things that aren't too caustic. And then the other thing you can do is sometimes the doctor will prescribe some antibiotics to get those bacteria under control. Once they get under control, sometimes it makes a huge difference. And we'll talk about these guys more when we do the skin. Are we okay? Is everybody sort of got that? Right? So, now you're looking at this. Oh, man. So this is pretty straightforward and simple. We'll kind of talk about the parts, and then we're going to talk about how we use these guys. So this is how we classify our exocrine glands according to their structure. And so we have two basic shapes that are here. This shape right here, this is considered to be a tube. So it kind of looks like a test tube. If we take a look at this shape over here, they talk about this guy being flask-shaped. And if you haven't seen a flask in the chemistry lab, it may not make it sense that it's flask-shaped. So the other word we use is vase-shaped. Vases are big on the bottom, and they're on neck for flowers. And so this is an alveolar structure, or the other word we use is acinar. Both of these words get used interchangeably these days. Are you with me so far? So I've got two basic shapes. What are they? Tubular. Alveolar or acinar. Either one of those terms are work. Two basic shapes. So what we do then is take the shapes, and we use those shapes to help name them. And then we talk about how many branches that we have. And so in this case, I've got one little tube, so he's called simple tubular. Pretty straightforward. And this picture is right out of your book, so these are all here. And so an example of this guy is what we call an intestinal gland. So in our intestines, remember I said that we were producing a lot of enzymes and mucus and stuff there. So if we had columnar cells here, these guys would go down into our intestines and come back up, and they would be producing a bunch of that stuff. So intestinal glands. This guy right here, notice, has a big curl in it. So he's got a coil, but he's still one tube. So he's called simple coil tubular. So this is the American sweat gland. So if you're an American, how are you releasing your secretions? Through exocytosis. All right? And so that's telling you how he releases his secretion. Now, this guy over here, he's got branches, but he's still called simple, and he's got the tube shape, so he's simple, branch, tubular. Now, we'll talk about the difference between simple and compound in just a second. So gastric glands, mucus glands, your esophagus, tongue, and duodenum, they're all simple branch tubular guys. Questions? All right, going to the next one. We've gone from our tube shape to our acinar alveolar shape, and so we see this little simple alveolar guy, and it says not found in an adult, a stage of development in the simple branch gland. So would you see these guys all by themselves in any of us?
No.
No, we might see them as they grow into these guys, but not just sitting there doing their thing in normal adults. So it's a transition kind of thing. You know, my buddy from South America where he used to work would get really upset if we talked about that because it's a transition stage. So this guy is called simple branch alveolar. So the shape is alveolar and simple branch because what happens is this neck, just like with this neck, all of the branches or the different alveolar shapes, or in this case tubular shapes, they all connect where that opening is. So that's why it's called simple branch, because everybody's connected to that one common opening. Okay? Yeah, I like that. Okay. Make sense? All right, so what do you notice about the difference here? There's a lot of them. Yeah, what? Cool. This guy's connected, right? And that guy's connected, and that guy's connected. But what about those guys? No, they're not directly connected, right? They're connected because they're connected to this guy who's connected here. That's what makes these guys compound. And so we just go back to our same naming system. Definitely compound because of all the branching. They're all shaped like tubes, so compound tubular. And so examples, mucus glands in your mouth, bulbourethral glands. These are the guys that when males get excited, but they're haven't ejaculated, but we need to clean out the urethra. This is the fluid that actually gets produced. And then the testes. We have these little tubes called seminiferous tubules where we make sperm, and the sperm can't be in a dry environment, so we give them a little fluid to live in, and these guys that's the kind of gland that's producing that fluid. So these guys have an alveolar or acinar shape, right? Multiple branches going all over, so compound, alveolar, and this is your mammary glands. And what kind of secretion is that again? African secretion. Good. So this guy is compound tubulo-alveolar, or sometimes we call him compound tubulo-acinar, because he has both shapes, right? And compound because that guy's on the common opening, that's on the common opening, that's on the common opening, but that is not. So compound. And then salivary glands, respiratory passages, your pancreas. Your pancreas is dominated by these things, and the pancreas we call them pancreatic acini, because they're actually tubular and alveolar, but when we look at them, we see a lot of these, so we call them acinar cells in the first cell.
So are the alveoli and the lungs named for the alveolar shape?
Mm-hmm. Yeah, that rounded sort of shape, exactly. Mm-hmm.
When it comes to learning these, do we need to be able to recognize all the areas that they're in, or just know this one for each of them?
Oh, the $1 million question. And that's where I was heading next. We planned that. That was a segue. You got a TV
Wait, not doing the test.
Good job. So what happens with these guys is we're back to, once again, that idea that learning works best if you have some meaning and some references for what you're learning. And that allows it to go into long-term memory easier. If not, we have a tendency to take those things and shove them into short-term memory and forget them. So here's what we really need to know. We need, if we were looking at one of these guys, we would need to be able to identify what it is. And so that's pretty straightforward, and we've gone through all the steps of that. If you were asked to draw one, so if I said draw a compound alveolar gland, you'd just be able to draw one. As long as we could figure out what it is, you're okay. You don't have to be very hard. What we'll do now is you learn the secretions, so you know what they are. and we've learned the basic shapes of these guys. Whenever we get to a gland, at that point, if we're in a system, and during that system we talk about one of these glands, we would expect that you now know where that gland is located. And so as we go to each system, you'll learn more and more locations of these different glands. But you don't have to sit here and memorize these lines. I mean, bulbourethral gland, chances are about 90% of us have never heard of this guy, so we don't even know where he's located, but have to go look it up why. We'll do this guy a lot in 233 reproduction. Does that make sense? So, next chapter, we'll talk about American sweat glands. So, by the time we get to that chapter, from that point on, you should know that the American sweat gland is a simple oil tubular gland, and put those together. Okay?
You want us to identify them visually, maybe
Or draw
name them, but not have to give an example of where they're located yet.
Not until we talk about it in a particular location. So what you're going to do is we are going to build on our learning. We're going to start with the foundation. If I know what it looks like, okay, if they tell me it's a compound alveolar gland and it has an amacrine secretion, then I can, in my head, see how it's secreting. But you wouldn't have to know it's in the mammary glands in this case until we get to 233 when we do the reproductive system. Are we okay? All right, so what you're going to find with Martini then, and you started reading chapter 5, so you probably noticed this already, is Martini will start, some of you just laughed, okay. You're going to read chapter 5. And so Martini and most AMP books, what they'll do is they'll lay the foundation by telling you what structures that system is made of. And they'll give you a brief little overview of what that system does. And then they start taking a look at all the components of the system. And they start looking at the components of the system from a tissue level up. And so they build that tissue or those structures for you. And so as you read these descriptions, and so it starts talking about the skin, and they'll say there's 3 layers of the skin, and they'll say the upper layer of the skin is made up of stratified squamous epithelium. Are you with me? You've already learned what that tissue is, you've got a picture of it in your brain. And then it'll say stratified squamous epithelium is divided into 5 strata. And he'll start talking about each stratum. And because you kind of had an image of stratified squamous epithelium, when he says the innermost layer is the stratum basal, and he says it's made of a single layer of cuboidal cells, you can think back to tonight when you're in the lab and go, oh yeah, I remember seeing that. And then he'll have a picture of it. See how this is going to work? And so we're going to constantly build on what we learned from week to week and day to day. And so to help facilitate that, we're not going to try to fill our brains up with stuff that we're not going to use reasonably soon, because you're not going to remember it anyway. So we should focus on the things that we need to know right now so we can keep doing our building. Right? I know it's a lot of brain theory stuff, but trust me, it actually works. And if it doesn't, then you should not have to memorize this much right away. So that was a joke. So we finished with epithelial tissue. The next tissue is connective tissue. And so this is basically how we break these guys down. You have connective tissue proper, fluid connective tissue, and supporting connective tissue. So when we take a look at connective tissue proper, we divide it up into 2 groups: loose connective tissue and dense connective tissue. And the real, terribly complicated scientific way that we describe these guys is everything here is real loose and everything here is real tight. And that's exactly how they just look at the structure of the fibers themselves, and then, these guys are all over the place, and these guys are tight real back together, and so we'll just call them loose and dense connective tissue. And that's part of what we call connective tissue proper. And then these guys are fluid tissues because they flow. And so blood and lymph connective tissue, so both of these guys flow. And then supporting tissues, one of the things that dominates these guys is when we take a look at the matrix, it's a really, really, really firm matrix. It's dominated by fibers and cells, and has very little ground substance or fluid portion of these particular connective tissues. So if you taking a look in your notes, where it says connective tissue, it shows you the functions. It just talks about the structural framework for your body. Obviously, it's blood and lymph, it transports stuff. It's going to protect your body. Obviously, bones and connective tissue. And everywhere we find some really important kind of internal structure, we try to the best of our ability surround it with a bone. There's some limitations to that. Because if we made you this really hard case and really solid bone, this is how you'd be. You'd have to walk around like this all the time, because all your internal organs would be solid bones, and only your arms and only your legs would move. So you couldn't bend over when you drop your keys, and you couldn't sit in a chair. So we got to have it going pretty like this there. And so support and that kind of stuff stores energy. Who's going to store energy here? Somebody said fat. Yeah, fat is adipose tissue, and adipose tissue is a loose connective tissue. And so fat is going to be this. And then within the body are invading microorganisms, and what we're going to find if you take a look at these tissues, we've got a lot of cells associated with the reproductive, right? The immune system that's going to help take care of us with the protection kind of idea. So when we take a look at this tissue, we need to kind of classify him a little bit. So one, he has to have some cells that are going to be present. That's what makes it a connective tissue, and there's some specific kind of cells that we see in some, and then some of them have a lot of different cells. The second thing you have to have is this tissue is going to produce fibers. This tissue always has a fiber component, and it will have large numbers of cells called fibroblasts that are dedicated to doing that particular process, which is creating fibers. And then the third thing we see with this guy is he's going to have some non-living kind of fluid that surrounds the cells, and we call that the ground substance. When we combine the ground substance and the fibers together, and get that cellular component too, but mostly the fibers and the ground substance, we're going to refer to that as the matrix. And the matrix is going to contain the cells. And we said the cells are embedded in the matrix. Okay? The matrix is going to be real variable with these guys. One of the variables will be dependent on the ground substance. If you have a really watery, fluidy ground substance, then the matrix is more fluid. And if it's almost non-existent and there's very little fluid that's there, then it's going to be really, really hard in front. Does that make sense? Okay. So, on the next part of your notes and in your textbook, and that starts on page 121, they show you a bunch of different cells. And so we're going to zip through all these cells and talk about them a little bit. When they show you this picture right here, this is actually a nice picture or slide of areolar tissue, which is a loose connective tissue. And areolar tissue has a lot of different cells in it, depending on where it's located in the body. We'll talk about them more specifically. So what they have a tendency to do in textbooks is they show you this picture, and they show you a picture of something like this. And what they do is they just throw all the cells in here. We might find a connective tissue as this big sort of generic representative sort of connective tissue. So that's what they've done right here. So what we're seeing here is collagen fibers here. We see elastic fibers here, we see reticular fibers here, and those are our three kinds of fibers that we have. And then they're throwing in all kinds of cells that are in here. So up here they're showing you a melanocyte. Melanocytes, though, are only found in connective tissue of the skin. And so, and actually up in the epidermis. So you don't really find them all over the place. And then we see some other guys that are present here. So rather than spend a lot of time just looking at this picture and talking about these guys, we'll kind of run through them and take a look at some pictures of them. So this guy is called a fibroblast. And so when you see the word blast, that means to build or to create or to synthesize or to make. And then we put a term in front of it, a prefix, to tell us what it's making. So fibroblasts make fibers. What happens is fibroblasts can divide, and then they mature, and they become a fibrocyte. And the fibrocytes sit around and just make these fibers to help maintain the homeostasis and health of the tissue that we're talking about. And so this is a real typical looking group of fibroblasts here. They would be making collagen fibers, reticular fibers, and elastic fibers, because those are our three fibers of our connective tissue. So that would be their role in any connective tissue. So this guy's called a macrophage. He's one of the defense cells that you have. And macrophages' job are to go through phagocytosis. And so, did you guys talk about that as 11? Okay.
Sounds familiar.
Yeah, where you put out the extensions and you grab something and stuff in it. It's probably in endocytosis, and we talked about moving stuff in and out of the cell. So what they do in those pictures a lot is they kind of show you these two long kind of stubby extensions that grab something and pull it in. And this is actually what it looks like in the real cell, but you can see these extensions going out, grabbing on this guy, they're pulling in, this guy's being pulled in. We've got pseudopods going out here pulling this guy in. This guy's halfway in already. These guys are toast, they're almost inside the cell. And so this guy's busy engulfing all of these guys. Once we get them inside, we just match, so they're now in a vesicle, right? Because they'll be surrounded by some membrane. We'll take that vesicle and move it over to where a lysosome is, and the membranes will fuse together. The lysosome is full of digestive enzymes, and these guys will be broken down and destroyed to protect you. Eventually, these guys will engulf, and they're going to get to the point where they're worn out, and then they just start to die, and they either fall apart, and other macrophages come in and eat up their parts, or the big macrophage comes in and just eats that guy up. So these kind of how it works for these guys. This is a adipocyte. So when we see site, that means cell, and adipo from adipose. And so this is a cell that makes up your adipose tissue, or real bluntly, this is a fat cell, and this is fat. And so when we take a look at this guy, once again, this is one of the few cells that you have in a human being that's dominated by a vacuole. And this vacuole, remember, this is a cell, and cells are tiny. And so this vacuole is full of an oil droplet. So just one little droplet occupying this entire cell. And so when you take a look at this guy, the vacuole is so big, it pushes the cytoplasm out to the side of the cell. So this dark streak that you see, this is where all the cell machinery is at. And this is all just a gigantic oil droplet or fat. And the nucleus gets shoved way over here and kind of bulges to the outside a lot. So when they first saw these guys, they weren't really sure what they were, what kind of tissue it was. And because they had this big bulging nucleus that you see here, and these are little small vessels here, the big nucleus that's here, they reminded them of the king's ring. So the king used to wear this big ring, and it stuck way up. And when he wanted to seal a special letter, he put some wax on there and stick the ring into the wax, pull it out, and it had his symbol in there. So that was his signature, and it was called a signet ring because of that. And so these were called signet ring cells because of this guy that stuck up. And then at some point somebody figured out that fat cells, and they were then called adipocytes after that. So, what? Venules. Remember that first week when we did the circulatory system? And we did arteries to capillaries to veins and the heart thingy. Well, that was a little oversimplification. So we go arteries to arterioles, arterioles to capillaries, capillaries to venules. So venules means little tiny veins. And little tiny veins, venules, collect into larger veins that then go back to the heart. So these are little tiny guys that are little tiny like capillaries would actually go into to take the blood away.
They're feeding the fat cells.
Yeah, but in this particular case, when they did the slides, they have to cut through venules. So they're helping, with feeding in this case. These guys are taking the blood away. So they're moving the waste products and CO2 out of the tissue because the arteries, arterioles, and the capillaries brought it to the tissues. Is how that works. Are we okay with adipocytes? And the job of fat is to? What? Store energy. Anything else? What? Protection. Somebody said the I word. Insulation. So do we want to have no fat?
No.
That's bad news. Our joints need to have fat, the greater omentum that sits underneath the muscles of our abdomen, not on top. Because the greater, you don't necessarily want a lot on top, so they can't see your six-pack. So, but the greater omentum underneath is there to protect the abdominal organs, so we need some fat that's present there. It needs to be in your skin as a form of insulation. So a certain amount of fat is important. You just don't want your percent body fat to be too high. And so you can't have no fat in your body. That's not a good thing. Because these guys are also your source of energy reserve. So, if you were starting to starve to death, we're going to need these guys to kind of help us out. These guys really don't regenerate themselves, they don't go through mitosis. And so when they first, I think the book sort of talks about this, they first got liposuction, they thought this was going to be cool, because you'd suck the fat out and you'd be good to go. And it turns out that there are stem cells associated with the connective tissue where these guys are at. And so the people didn't change their diets, and they thought, well, I sucked all the fat out, I don't have any bulges in